Good afternoon and a very warm welcome to all. So we have finished the topic microbial metabolism. So today we are going to start with a new unit, that is culture media. And today we can discuss what are the common ingredients used in a culture media. Introduction. The culture media or growth media, it is a liquid, either a liquid or a gel, which are designed to support the growth of microorganisms. And there are different types of media suitable for growing different types of cells. Here we will discuss microbiological cultures used for growing microbes such as bacteria or yeast. One of the most important reasons for culturing the bacteria in vitro, it is its utility in diagnosing infectious disease. Isolating a bacterium from the sites in body normally known to be sterile, it is an indication of its role in the disease process. So, isolating the bacteria from the sites of the body which are normal, known to be sterile. Culturing bacteria, it is also the initial step in studying its morphology and identification. So bacteria have to be cultured in order to obtain antigens from the developing serological assays or vaccines. Certain genetic studies and manipulations of the cells also need that the bacterium should be cultured in vitro. Culturing bacteria may also provide a reliable way estimating their numbers. That means viable count can also be taken using the culture media. Culturing on solid media is another convenient way of separating bacteria from the mixtures. So coming to the history, the Louis Pasteur used simple broths which are made up of either urine or meat extracts. And also the Robert Koch realized that the importance of solid media and used potato pieces to grow agar. So, it was on the suggestion of the Fanny Elshemius, wife of Walter Hesse, who was an assistant to Robert Koch, that she had a suggestion that agar was used to solidify sulfur media. Because with the use of the agar, attempts were made to use gelatin as a solidifying agent. Gelatin had some inherent problems. It is not capable of solidifying. It exists as liquid at normal incubating. So it exists as a liquid even though when we are normally incubating temperatures, and was digested by certain bacteria also. Next is composition of culture media. The bacteria which are infecting human beings are chemoorganotrophs. When culturing media, when it is very important to provide similar environment and nutritional condition that exists in its natural habitat. So, the natural conditions can be provided by culturing media. Hence, an artificial culture media must provide all the nutritional component that the bacteria gets in its natural habitat. So, while we are taking the artificial culture media, we should know how much nutrients are available for the bacteria from the nature, etc. Most often, a culture medium contains water and a source of bread. So what happens in this condition is that it may provide culture medium has contained water, source of carbon, energy source, nitrogen, trace elements, etc. And apart from this, the pH of the medium must be set accordingly. Some of the ingredients in the culture media including water, peptide, casein hydrolysate, meat extract, wheat extract and malt extract. These are the different components which are present in a culture media. Now coming to the common ingredients in culture media. First one is water. Water, it is an essential for the bacterial growth and it uses deionized or distilled water. Next is peptone. From hydrolyzed animal. Peptone can be obtained from the hydrolyzed animals or plant proteins and it provides nitrogen and amino acid. Next is about meat extract. It may provide amino acid, vitamins, mineral salts, that is phosphate and sulfate. Yeast extract, it is used to stimulate the growth of bacteria. Stimulation of the bacterial growth. Mineral salts, traces of mineral, potassium and iron and calcium which are essential for the enzymal biotic. Essential for the bacterial enzyme activity. So, the mineral salts are also present in the culture media which is available in trace amounts of potassium, magnesium, calcium, iron and calcium. These are the mineral salts essential for the enzyme activity within the bacteria. Next is carbohydrates. The carbohydrates provide bacteria with energy and carbon source. Next coming is agar. It is an inert polysaccharide from seaweed or marine algae. It is originally solidifying agent which carbohydrate of it is a solidifying agent with a concentration of 1 to 2%. So it can be asked as a viva question, what is the concentration of the agar in the media? Solid media agar concentration, that is 1 to 2% dissolved at 92 degrees Celsius solidified. So it dissolves at 90 to 100 to 45 also. Next is about the formulation of all the plants and different kingdoms into different roles or functions. First one is nutrients. It can be either proteins, peptides and amino acids are coming under nutrients. Next is it provide energy. That means carbohydrates are provided. The next is essential metals and minerals. Example, calcium, magnesium, iron, trace metals and phosphates and sulfates. Buffering agents. The buffering agents are actually the phosphate, acetate, etc. It does not move towards the more charged area. It remains within the silent. Selective agents used in the culture media, chemicals or antimicrobial agents, etc. So what do you mean by antimicrobial agents? It is the gelling agent usually used in agar. There is often an overlap of conditions of some media components. Thus, the protein hydrolysates will support the amino acid nitrogen. So this is often overlap of function of some media components, and this protein hydrolyzes and it supply amino nitrogen energy. Some minerals or metals acts as a buffering agents. Phosphate buffers are important suppliers of minerals and nitrogen. Next is about nutrients. So coming each and every points we can discuss. Nutrients, a group of bacteria are now defined as chemoorganotropes, required amino nitrogen compounds, and these are essentially from her growth. Meat infusions contain water-soluble fractions of proteins. Proteins means amino acids and small peptides along with the water-soluble products such as vitamin, trace metals, minerals and carbohydrates. Such infusions or extracts may have been regarded as peptones, but their amino acid nitrogen content was usually too low to sustain the growth of or the number of bacteria. It was not still deliberately attempts were made to hydrolyze proteins, the acids or enzymes that essentially so that sufficiently high concentration of water-soluble protein fractions, that means front side, which made. So these are all about nutrients. The next is the most common substances added to the culture media as a source of energy to increase the rate of the organism is mainly what? Glucose. Glucose acts as the energy component. Other carbohydrates may be used other than glucose. So carbohydrates added to the medium at 5 to 10 grams per liter are usually present in biochemical substrates. So the carbohydrates added to the medium at 5 to 10 grams per liter are usually present as biochemical substrates to detect the production of specific enzymes in the identification of organisms. It is used to add pH indicators to such formulation. So next is the big slide. It is important that pH of the medium is proposed by the home. So the culture media, it posts the culture media and the growth of the desired microorganism that. So in this condition, so there is a it is important that pH of the culture medium is poised around the optimum necessary for the growth of desired microorganisms. There are certain buffer compounds at specific pH values, especially necessary when fermentable carbohydrates are added energy sources. Next important ingredient is phosphates. Acetate, citrate, zitrione compound specific amino acid phosphates are examples for buffering agents that may also be added to the culture media. A side effect of such compounds is the ability to chelate divalent cations and magnesium. The polyphosphate salts sometimes present in the sodium phosphate are components which can bind essential cations so many that they are made inaccessible for the microorganisms. So what are the different side effects of such compounds? Is their ability to chelate divalent cations. Polyphosphate salts sometimes present in some solid, sodium solid, etc. and they are made conformly to the formulation. One specificity in a medium having separate elimination. So effect of this binding can be seen in the diminished growth of failure to grow at all. And so these are all. Then opacity formation in the medium after heating or standing is commonly caused by phosphate interaction with metals. Such phosphate precipitates can very effectively be bind and over the nerve effect. So this may result in what? So this phosphate